Hello dear students. In this lecture we are going to see about the mechanism of unoxygenic photosynthesis. In our last lecture we have seen about the oxygenic photosynthesis which is carried out in cyanobacteria and plants at the thylakoid membrane. So that oxygenic photosynthesis, it is carried out in two ways, cyclic and non-cyclic. So if electrons it reverts back, it is known as the cyclic process, but if it is a linear, if it is a Z scheme, or yes, if it is a linear, then it is termed as the non-cyclic oxidative photosynthesis or oxygenic photosynthesis. So that we have shown that it consists of two photosystem 1 and photosystem 2, and at this thylakoid membrane how the electrons flow, as there is when the electrons they passes, or when there is flow of electrons, there is pumping of H plus ion, then this H plus ion get accumulated or are driven into the ATP synthesis which generates ATP. So this already we have discussed in our last lecture, that in oxygenic photosynthesis there is production of NADPH and ATP. Now in this lecture we are going to focus at the unoxygenic photosynthesis mechanism in purple bacteria. Okay, so that will be our main focus. Unoxygenic mechanism, as there is no oxygen produced in this process, hence it is termed as unoxygenic mechanism, means no oxygen. Example of unoxygenic photosynthesis are purple bacteria. This purple bacteria, they can or they accept electron from sulfur or hydrogen sulfide or any hydrogen atom to reduce atmospheric carbon dioxide into the organic compound. So that is the main theme. If we compare the oxygenic photosynthesis, then in oxygenic photosynthesis, electron donor was water. Okay, water was splitted into hydrogen and oxygen, and then it serves as the electron donor. But instead of water, in unoxygenic mechanism there is sulfur or hydrogen sulfide or any hydrogen compound which serves as a electron donor. Okay, they serve as a electron donor. So this thing you have to keep in mind, that is the main and major point of difference between oxygenic mechanism and unoxygenic mechanism. That in oxygenic mechanism, water along with the light serve as a electron donor, but in unoxygenic mechanism, sulfur or hydrogen sulfide or hydrogen compound, they serve as a electron donor to reduce atmospheric carbon dioxide and produce organic compound. Okay, so that is the main point to remember. So depend on the type of electron donor, like sulfur or hydrogen sulfide or hydrogen compound. So depend on the type of electron donor, we can divide purple bacteria into two types. Okay, we can divide this purple bacteria into two types. So that two types are purple sulfur bacteria, that is PSB, and purple non-sulfur bacteria, that is PNSB. What is PSB, PNSB? What are purple bacteria? In last videos we have shown about the examples of this photosynthetic bacteria. Okay, so in that we have discussed about this purple bacteria, green bacteria. So here we will see how actually flow of electron is being carried out. Okay, so depending on the type of electron donor, we divide purple bacteria into two types, purple sulfur bacteria and purple non-sulfur bacteria. In case of purple sulfur bacteria, this bacteria can accept the electron from hydrogen sulfide or sulfate atom. Okay, it can accept the electron from H2S or sulfur atom in purple sulfur bacteria. And in purple non-sulfur bacteria, it can accept electron from hydrogen or any hydrogen compound. Okay, that is the difference between this PSB and PNSB. In PSB bacteria, they can accept the electron from H2S or sulfur, and purple non-sulfur bacteria can accept the electron from hydrogen or any hydrogen related compound. Okay, that is the difference between PSB and PNSB. But the mechanism of flow of electron is same, just the donor is being changed. Okay, as the donor is changed, definitely the acceptor will also change. Okay, so the mechanism of flow of electron is same in both PSB and PNSB. So second point, what you will keep in mind, that in purple sulfur bacteria can accept the electron from hydrogen sulfide or sulfur, and purple non-sulfur bacteria can accept the electron from hydrogen or any hydrogen compound, and mechanism of flow of electron is same in both PSB and PNSB. Okay, so that is the second point to keep in mind. Next, if we speak about this flow of electron, then photosynthetic reaction centers, same, it begins with the reaction center as we have shown in oxygenic mechanism. So photosynthetic reaction centers, these are surrounded by the antenna pigments, and we know the function of antenna pigments. They capture the light and transfer it to the reaction center. So same is the case in Unoxygenic, like oxygenic mechanism. In this also, reaction centers are surrounded by the antenna pigment, so that antenna can capture the light and give it to the reaction center. Okay, so as this light is being transferred to the reaction center, it gets excited. Okay, it reaches to the excited state, and that excited state is given by the mark. Let's see the diagram. Okay, so this is the mechanism of flow of electron in purple bacteria. Okay, and as I told, the mechanism is same in both PSB and PNSB. So yes, we know that these reaction centers are surrounded by the antenna pigments that capture the light and transfer it to the reaction center. Now your reaction center is P870. Okay, P870 is the reaction center, means what? It can absorb the light at a wavelength of 870 nanometer. Okay, so red or infrared light is been focused or is been entered into this reaction center with the help of this antenna molecule. As the light passes, okay, as the light passes, there is excitation. It reaches to the excited state. But here not only light is sufficient to excite this reaction center. As in oxygenic, we have discussed that just light is not sufficient to excite. We even require the water. Okay, and that water is split into hydrogen and oxygen, and then this oxygen it is involved in the transport of electron, or it serves as the electron donor. Same is the case with this purple bacteria, but instead of water, there is involvement of hydrogen sulfide. Okay, H2S, there is involvement of hydrogen sulfide. So along with the light, here hydrogen sulfide act as a electron donor. So light is been passed, or light is been transferred to the reaction center along with the hydrogen sulfide as a electron donor. Okay, and then it get excited. Okay, this ground state gets excited, and then electrons are transferred step by step to different components. But before seeing this transfer process, let me tell you about this volt. Okay, this reduction potential scale. This is known as the reduction potential scale. Okay, it is the reduction potential scale which is measured in volt. Now here if we'll see downside, then it is termed as the poor electron donor. Okay, it is the poor electron donor. Now means what is actually the theme? This complete process is based on, or it relies on, reduction potential. Okay, it completely relies on reduction potential. Suppose I have two molecules, molecule A and a molecule B. Okay, so this A, if it is in the reduced form, when I'm telling reduced form is what? It has the electron, it consists many electrons, and it have the ability to reduce the other compound. Okay, if A itself is reduced, it can reduce the B compound. If B is reduced, it can reduce the C compound. Okay, so the main theme is, if high reduction potential passes the, means overall the concept is like that, high reduction potential can pass the electron to low reduction potential. Okay, so if A is in the reduced form, it can reduce the compound B. B can reduce the compound C. Reduce means what? It have the electron. It have the set of electron. It is fully occupied by the electron, and it will donate the electron. Okay, that is the reduction potential. It can do anything. It can accept the hydrogen. Okay, depend on the theme. It can add or remove the hydrogen. Okay, so that is the main concept of reducing power or reduction potential. So here if we'll talk about the plus sign, then plus, it indicates that poor electron donor. Okay, it is the poor electron donor, means it lacks the electron. It doesn't have the electron. It depends on the other system. So it depends on the hydrogen sulfide compound or light, so that this light and the hydrogen sulfide, it gives the electron. As it receives the electron, it get excited and reaches to the negative value. Here negative value indicates that strong electron donor, means what? It is rich in electron. Now it can transfer the electron to different components. Okay, so here this is the highest electropositive reduction potential. Okay, poor electron donor का मतलब ही होता है higher electropositive reduction potential. Okay, because it is the highest positive value, means what? It can accept the electron. Okay, so this poor electron donor means what? It is nothing but highest electropositive reduction potential. The scale is of reduction potential, so I am using the word reduction potential. Highest electropositive reduction potential. And this negative value, that is the strong electron donor, means what? We can also say that it is highest electronegative reduction potential. Okay, here highest electropositive, here highest electronegative. And positive means what? It can accept the electron. As it accepts the electron, it become rich, it can donate. Okay, it get excited, and from this it can donate to different components. Okay, so negative means it can donate, and positive means it can accept. Okay, so that is overall theme. And even this third point also you have to keep in mind, because the base of this mechanism is completely depend on this reduction potential. Okay, and this electropositive and electronegative value. Okay, so we were here that it become excited. Okay, excited means now it is rich in electrons. Okay, so as it is rich in electron, it will donate the electrons to different components. So from P870, the electrons are transferred to the BPH. Okay, that is nothing but the BPH, that is nothing but it stands for the bacteriophyophytin. Okay, bacteriophyophytin, and after bacteriophyophytin, electrons get transferred to bacteriochlorophyll. Okay, here bacteriochlorophyll is also there, but it is not mentioned in this diagram, but it is present. Okay, so this bacteriophyophytin and bacteriochlorophyll, these are the unique components which are not present in the oxygenic mechanism. Okay, that is the difference between oxygenic and unoxygenic mechanism. अगर आपको ऑक्सीजेनिक आपने ऑक्सीजेनिक का वीडियो देखा होगा तो आपको क्लियर हो जाएगा कि यहाँ पर सिर्फ क्लोरोफिल पिगमेंट इन्वॉल्व है, लेकिन यहाँ पर इंस्टेड ऑफ दैट क्लोरोफिल देयर इज इन्वॉल्वमेंट ऑफ बैक्टीरियोक्लोरोफिल BCHL and bacteriophyophytin. Okay, so here BPH is there और उसके बाद bacteriochlorophyll भी है which has not been mentioned but it is present. Okay, so these are the photoreactive centers in oxygenic unoxygenic pathway. Okay, so that is the difference between this, and these are the light harvesting molecule. BPH or BCHL means what? Bacteriochlorophyll and bacteriophyophytin. These are the light harvesting molecule which can gather the light and can excite and transfer the electron. So different reaction centers are present in the cell membrane. So there here P870 is one reaction center, bacteriochlorophyll is another reaction center, bacteriophyophytin is another reaction center. Means what? They can harvest the light and they can transfer the electron. That is just the main function of this light harvesting complexes. From this P870, electron get transferred to bacteriophyophytin. From bacteriophyophytin, it get transferred to bacteriochlorophyll. Okay, and in this system, some are embedded carriers, some are embedded electron, and some are mobile carrier electron. Okay, so this cytochrome C2 and cytochrome BC1, these are the embedded. Okay, and quinone molecules, these are the mobile carrier, means they can transport the electrons. Okay, so from electrons get transferred from this excited state to bacteriophyophytin, from bacteriophyophytin to bacteriochlorophyll, from bacteriochlorophyll to quinone complexes. Quinone, these are the mobile carrier. So quinone A, quinone B, and then to the quinone pool. Okay, from quinone pool, these electrons are transferred to cytochrome BC1. From cytochrome BC1, it gets transferred to cytochrome C2. From cytochrome C2, it is back to the reaction center called as P870. Okay, so this is the cyclic process. And as these electrons are moving, it generates a gradient. Okay, it generates a proton motive force of H plus of proton. Okay, hence it is known as the proton motive force. So as these electrons are moving, it generates a electron motive force which produces the ATP and NADPH. Okay, so here light harvesting molecules, they excite the light. Photopigments can harvest the photon and they can be excited. Okay, they harvest the light, they get excited, electrons get transferred, and after exciting, slowly they become down and they reaches back to the ground state. Okay, so this they come to the ground state. This energy pumps the H plus from inside to the outside of the cell. Okay, this flow of electron, it generates the energy and it pumps H plus from inside to the outside of the cell, which creates the proton motive force, high density gradient outside the cell, and then they can pump the H plus down the cell, which will help to produce the ATP. That is overall concept, overall view of this unoxygenic mechanism in purple cell, in purple bacteria. Okay, both sulfur and non-sulfur. Now here the main thing is, as the hydrogen sulfide, they serves as the electron donor. Okay, hydrogen sulfide, they serve as the electron donor. So this hydrogen sulfide will get reduced to, or hydrogen sulfide is totally reduced form, and sulfate ion get oxidized. In short, I want to tell just one thing, that here at last sulfate ion are produced. Okay, at last sulfate ion are produced, and this sulfate ion, it cannot leave its own, it will react with the water and produce hydrogen sulfide. Okay, or it will produce sulfuric acid, H2SO4. So from H2S it starts, H2S produce the SO4 combined with the water and produce H2SO4. That is the concept behind all this purple bacteria, overall this purple bacteria. Okay, we will revise once again through this diagram, actually where this flow of electron takes place. Okay, so it takes place at the membrane. Now we know that in purple bacteria, or in any category, purple sulfur bacteria or purple non-sulfur bacteria or photosynthetic bacteria, the process of photosynthesis is carried out at the cell membrane, or we can say at the chromatophores. Okay, as I told earlier, we have discussed that in eukaryotes different site is there, in prokaryotes different site is there. Okay, so this reaction centers, these are embedded in the cell membrane. Different reaction centers are embedded in the cell membrane. So here it actually carry out. These are the LH, are light harvesting complexes. Different light harvesting complexes are present, like pH70, bacteriochlorophyll, bacteriophyophytin. Okay, so these are the light harvesting complexes. Light enters. Okay, light enters here. So as light enters, by mistakenly I told that hydrogen sulfide is provided here, but hydrogen sulfide it gets here because it is the strong electron donor. Okay, so here light is sufficient to excite it, and here hydrogen sulfide can act as the strong electron donor, which can donate the electrons to these different components. Okay, so that was one point. Okay, so light get fall onto these light harvesting complexes. Here antenna pigments are present. This antenna pigment transfer the light into the reaction centers. Okay, so it start from this reaction center pH70. As light passes, it get excited. Okay, pH70 star mark indicates that it is in the excited stage. Okay, so from pH70 it reaches to the pH 70 star, means what, from ground state it reaches to the excited state. Then electron get transferred to this bacteriophyophytin. From bacteriophyophytin, bacteriochlorophyll is also there. From that it get transferred to the quinone. Okay, this is the quinone pool. Quinone pool means different quinone molecules like quinone A, quinone B, and then to the quinone pool. So from quinone pool the electrons are transferred to BC1. Okay, bacteriochlorophyll 1. Then from this it get transferred to the, sorry, not bacteria, yes, BC1, bacteriochlorophyll 1. Then from this it get transferred to the iron sulfur complex. From iron sulfur complex to last cytochrome C2. Okay, and when these electrons are being flown, there is pumping of proton. Okay, and then again back to the ground state, that is reaction center pH70. So during this process there is pumping of H plus ion, and this complex, okay, which is also called as ATP synthesis. This complex drives the, as there is H plus ion concentration is high outside the membrane, it drives inside the membrane, and then there is generation of ATP. So this is all about the process of anoxygenic photosynthesis in purple bacteria, which involves the production of ATP. Now simultaneously NADH is also being produced. Okay, so how it is being produced, that we will see in our further lecture, which is interconnected with this lecture, and that will be the part 2 of this anoxygenic mechanism. I hope this will be helpful for you. Thank you.